Yes, Jinhee, thank you very much for the introduction. I would like to apologize that I do not speak Korean. However, I'm grateful for your patience with me so that we can share this great information with you. Thank you to all of Pile Korea. They are a wonderful group of people to work with, and they honor us by working so hard with us and you as customers. I'm sure Jinhee mentioned, my name is Jason Meyer. I apologize, my camera was not going to work today. However, obviously I can speak and present the material. The history that I have in biotechnology is well over 20 years in both scientific and business side. I've done quite a bit of work with molecular biology, next-gen sequencing, induced pluripotent stem cells, and now CRISPR. I've been very happy to see the power of the evolution of gene editing technologies. And I'd like to share our perspective with you and how CRISPR is going to hopefully really strengthen the way that we're able to do research and understand the genetic issues that we have and how we're able to affect them in the future. So I'm going to go over a brief historical review of gene editing, talk about specifically targeted gene editing. I'll also go over the power of CRISPR, how it is performed, and then ultimately what's being done with CRISPR. And of course, I'll always be happy to take questions at the end. So in studying gene function, before anything was targeted, a lot of what was performed was random. We relied on the power of probabilities for these type of edits in the genes that we wanted to study. And we look back initially, chemical mutagens and chemical mutagenesis was used in order to randomly introduce mutations in the genome. And these chemical mutagens that were used, were such like ethyl methane sulfonate, and N-methyl N-nitrosourea. These compounds, again, the living cells were exposed to these compounds, and hopefully, as you can see on the screen, we would see a nucleotide change and hope it would result in an altered gene function. This evolved into insertional mutagenesis, where either through potentially viral activity or with transposons, one or more base pairs of the genetic sequence would be removed, added, turned around, all these different options. And again, the scientists at the time were relying on the probabilities and statistics to be able to see that there was something in a gene of interest that they wanted to study. And that also relied on the ability of the scientists at the time to detect these edits. And deep sequencing of genomes wasn't really a possibility in great depth at the time. Later on, as technology developed, then we got into targeted mutagenesis. And I'm sure I'm not giving new information to many of you. At the time, creating plasmids with specific edits, and then were introduced into the cells, hopefully in the nucleus, where potentially these were used in a homologous recombination process to create a specific edit. And again, not necessarily highly controlled, definitely not able to specifically target a location in the genome. And again, trying to really figure out, was this where it was going to be? Was the edit that was introduced useful? Was there a way that they could get real value out of this? And I talked a little bit about the history. So I wanted to kind of point out, this is all kind of before 1990, 1985, a lot of these technologies were available, from the 1960s to the 1970s. Around 1985, then we started the era of being able to target edits in the genome. Zinc finger nucleases allowed us to do that. And that's around the same time that we began the Human Genome Project. Fast forward to about the year 2000, where the Human Genome Project is completed. And that was all done with historical sequencing techniques. And then in around 2005, 2006, next-gen sequencing emerged on the market, making it much easier to analyze what was going on, what was happening, what the gene sequences are, what type of differences were being noticed in the genomes. Zinc finger nucleases then kind of got an upgrade when TALENs were discovered in around 2010. Again, I'll talk a little bit more about this, but it allowed for a little more specificity. And then in about 2008 to 2012 time period, CRISPR-Cas9 editing became available. As I'm sure many of you remember, the CRISPR revolution started. And then fast forward to 2020, the year 2020, the Nobel Prize was awarded to Doudna and Charpentier in chemistry for the discovery of CRISPR-Cas9. And then, of course, the usefulness of that as a gene editing technology. Looking at these gene editing technologies, going back to the restriction enzymes, there are many options, relatively easy to use these. The restriction enzymes had well-defined sequences in DNA that they would recognize, and then they would be able to be cut. Those cuts would then be able to be utilized for either some type of edit or allowing a specific sequence of genes or DNA to be introduced to the genome. The issue with restriction enzymes, I'm sure you all recognize, is that there are many restriction sites in the genome. So targeting a specific location was just not possible. Fast forwarding to zinc finger nucleases. As you can see, these orange boxes on both the top and the bottom side here, they represent proteins that recognize specific nucleotides of the DNA. And these would basically get into the major groove of the DNA double helix. It would then be bound with these very specific nucleases that, when they were together, would create a cut. It was a very programmable system. It was kind of limited in the number of base pairs that it would recognize. And on top of that, even when you were able to get a good protein created, it wasn't always successful. You may have the right sequence of proteins for the right sequence of DNA, but the success rate was pretty low with these. And to do this type of protein engineering did require a lot of protein expertise and time and effort in your laboratories. The same situation with TALENs. You just were allowed to increase the recognition. I believe I have a typo here on my slide. I apologize. It made it much longer for the recognition of the DNA sequence. Obviously, it became a lot more programmable, more specific, but it still was limited in the fact that it required a lot of protein engineering to do this. And the success rate was still relatively low. And I'm sure many recognize engineering proteins and creating proteins is a laborious and longer process. But then we get to CRISPR, and CRISPR-Cas systems are very programmable. The longer recognition sites, for example, CRISPR-Cas9 has about a 17 to 23 base pair recognition location. And it also requires a specific signal on the DNA for the enzyme to cut. And it doesn't require any protein engineering. The proteins and the enzymes are very consistent. You can modify them for different things you'd like to do. But however, just for simple editing, the nuclease activity works very consistently. It does not have to change to target different locations on the genome. That's what the RNA does. And making RNA oligos may not be as easy as making DNA oligos, but it is quite a bit easier than doing large protein engineering. So I'd like to shift over into explaining how CRISPR works. If this is a review for you, I apologize. I'll try to make this very straightforward and understandable. The components that are required for CRISPR-Cas systems, the way it was in nature when discovered, it was a two-piece RNA guide and scaffold. So the CR portion here, CRRNA, was the guide sequence. There's about a 20 nucleotide sequence that was homologous to a target in the genome that you wish to edit near. A portion of this did overlap with the tracer RNA that you'll see here in purple. This tracer RNA is also what we call the scaffold. So the piece here would bind, this piece of RNA would bind with this alternate piece of RNA on the tracer RNA. And then that would also bind to the enzyme. This scaffold fit into a groove of the enzyme and held it firmly in place. And all three of these pieces were required in order to find the DNA sequence of interest and cut the DNA. What was learned is, obviously, with any system, the more pieces you have, the harder it is for all of them to come together to work effectively. So it was decided to add this linker loop between the CRISPR and tracer portion of the RNA. And we currently call this a single guide RNA. And this has drastically increased the efficiency that CRISPR has been able to experience, because now all you have to do is get this single piece of RNA bound with the Cas9 enzyme in order to do the editing. Once that all binds up, we call it a ribonucleoprotein, or as scientists are very happy to do, make acronyms, we call it an RNP. And when this binds to the DNA sequence of target, and there's also an additional sequence that is recognized, the protospacer adjacent motif, or the PAM sequence. This is the signal that tells the nuclease, the enzyme itself, to do the double-strand cut. So that's the basis of the molecular biology of CRISPR-Cas9. Oops, hit the wrong button there. So what can you do with this? Well, I think many people recognize on the left side here, you can do a knockout. So once this RNP binds to DNA, finds that PAM site, and this double-stranded break occurs, as you see on the right side of the screen, the cells themselves will repair with the internal repair machinery that's available. And we all recognize that this machinery is very random. It does not make a perfect perfect repair in the DNA. Sometimes pieces are added or pieces are removed. And we call those, if they're added, insertions, as you see this little red portion here, or deletions. I'm not going to count them, but this green portion is shorter than the original one. And it doesn't necessarily matter if there's a nucleotide inserted or deleted. So we've combined it into one term called an indel. And as long as it's not a multiple of 3 nucleotides, it will create a phase shift in the transcription of the DNA into mRNA. And that will, of course, make a missense protein, or it could potentially create an unnatural stop codon, and which effectively knocks out or eliminates that gene from the genome. So it cannot be effectively transcribed or translated into a functional protein. And knockouts are very valuable in studying, gene function and protein function, et cetera. And this process is wholly reliant on the system's non-homologous end joining, NHEJ process. Now, with being able to create a cut, we also understand that sometimes we can introduce a homologous damage repair template. We know that DNA will use templates to repair, breaks that occur naturally or due to environmental hazards. And we take advantage of that process. We'll put in a template that upstream and downstream of the cut site that we've designed. And then in part of that, we will change the sequence so that we can either put in a SNV, a single nucleotide variation, where maybe we change, say a glycine to a leucine, or we can put in potentially a tag, whether it's a small tag like an HA tag or a flag tag, or even larger tags like a fluorescent tag, if you want to see where this protein is going. So this relies on homology-directed repair, and as I'm sure you understand, the more pieces that are involved, the less efficient things become. So that makes things really challenging, because now not only does the RNP have to combine, so you need the RNA and the enzyme to come together. You need it to get to the right location on the genome at the same time that this template is available for it to create the exact repair that we want. So CRISPR is really powerful. It works really well, and when you read it, it looks like it's really easy, but as you realize, how just dense the cell nucleus is, it makes it really challenging to get that done effectively. And we like to say it's as much art as it is science. So what has CRISPR meant to the community? And I'd like to point out, I did a quick PubMed search specifically for the term gene editing. And I did this from between the years 2000 to 2012. I picked 2012. That was, it just happened to be the year Synthego was founded. And I choose that because we're one of the few, if not the only company that solely focuses on providing CRISPR type services. And then from 2013 to 2023, there was 14 times as many citations in 10 years versus the 12 years here. Just the pure number of studies that were going on in gene editing may not be solely due to CRISPR, but it appears to us at least that CRISPR made it so much easier for scientists to study and publish good data in peer-reviewed journal articles once it became available to customers or to scientists. And we as a company typically go out and we talk to people about what they're doing in their laboratories. And of course, it's really important to understand, what are the gene editing technologies that you as customers and scientists are using. And in our benchmark report volume 2, in both commercial and non-commercial institutions, CRISPR was the largest gene editing technology that was used. So not only is it very powerful in being able to very specifically target locations for editing, it's obviously available enough to be utilized efficiently. And of course, as you can see, the other ones that are out there, such as RNAi, it's not really a gene editing technology as much as it's a gene altering. It's affecting the mRNA that's being produced and regulating the ability of the gene to express, as opposed to actually removing it completely. And then, of course, as you take a look, talons and zinc fingers are a much smaller portion, and other technologies are also still being used. So what can be done with this? And of course, it's not just academic laboratories that can do it, but there are also many commercial companies out there that are utilizing it. As you can see in this image here, now obviously Synthego is in here as a service provider, but you'll also notice companies that are actually utilizing CRISPR as a therapeutic strategy. So, companies like Blue Rock are using it to potentially create disease therapeutics. And I took a look, and in 2020, I was able to determine that there were 51 clinical trials using CRISPR as a therapeutic. And I'm fairly confident that that number has grown. I just wasn't not able to find more recent year numbers. I apologize for that. And these diseases that were being targeted are some of the major ones, cancer being, at the top of the list. There's also quite a few that are out there. Beta thalassemia is being targeted, sickle cell disease, and many more. One area that I know is growing is the neuroscience area. The type of diseases that are Alzheimer's-like or Parkinson's-like diseases are being targeted. However, the commercial is not just at disease therapeutics, but also at diagnostic applications. During the COVID pandemic, the COVID-19 pandemic, there were a lot of people that were utilizing CRISPR as a way to detect pathogens. Both infectious and non-infectious diseases were being just diagnosed with CRISPR. Now, this works for, obviously, like a PCR assay where you have a specific sequence that's unique to your pathogen. It's also able to detect SNPs and deletions, so genetic diseases, say like during prenatal screening. And then I'm not going to get into it, but there's ways to detect, compounds that aren't nucleic acids that are affected by the presence of certain nucleic acids. So, ultimately, what I wanted to hopefully really impress upon you is that the power of CRISPR is already being used by so many people, but we still have so much more that we can do with it. There are so many more diseases, so many more opportunities to study different areas of the systems, the biologic systems that are available. And I really do feel that the limit is only based on the intelligence and the creativity of the scientists that are utilizing CRISPR. And with that, I would very much like to say thank you. I would like to open this up for any questions, and I really am grateful for the opportunity to speak with all of you.